On this day in history, April 14, 2003, the Human Genome Project was completed, a significant milestone in the field of genetics and molecular biology. This ambitious project, which aimed to map and identify all the genes that make up the human genome, began in October 1990 and involved researchers from around the world collaborating on an unprecedented scale. The idea of sequencing the entire human genome, which is the complete set of genetic information encoded in our DNA, was first proposed by Dr. James D. Watson, one of the co-discoverers of the structure of DNA, in the mid-1980s. In 1988, the National Institutes of Health, or NIH, in the United States, along with the Department of Energy, also known as DOE, launched the Human Genome Project, or HGP, as a joint effort to unravel the mysteries of human genetics. The project was an ambitious undertaking, as the human genome is comprised of approximately 3 billion base pairs of DNA, which contain over 2 billion, 25,000 protein-coding genes. The initial goal of the HGP was to create a map of the human genome, which would identify the location and basic structure of all the genes. This involved developing advanced techniques for DNA sequencing, data analysis, and computational biology. The timeline of the Human Genome Project can be divided into 3 parts: the pilot phase, the production phase, and the post-production phase. The pilot phase, which began in 1990, focused on developing and optimizing methods for DNA sequencing, as well as establishing data-sharing protocols and ethical guidelines for the project. During this phase, researchers made significant progress in improving DNA sequencing technologies and reducing the cost and time required to sequence DNA. In 1996, the project entered the production phase, during which large-scale sequencing efforts were launched. The production phase saw a rapid acceleration in the pace of DNA sequencing, with multiple laboratories and institutions contributing to the effort. In 2001, the International Human Genome Sequencing Consortium, which was a collaboration of several research institutions from around the world, published a preliminary draft of the human genome sequence, covering over 90% of the genome. Finally, in April 2003, the Human Genome Project was declared complete, with the publication of the final version of the human genome sequence in the journal Nature. This was a historic moment that marked the culmination of over a decade of collaborative research and technological advancements. The completion of the Human Genome Project had far-reaching implications for various fields of science and medicine. It has provided researchers with a wealth of information about the organization and function of human genes, as well as insights into the evolution and diversity of the human species. The project has also paved the way for advances in personalized medicine, as it has allowed researchers to identify genes associated with various diseases and develop new diagnostic and therapeutic approaches based on an individual's genetic makeup. Moreover, the Human Genome Project has led to the development of new technologies and techniques in genomics and bioinformatics, which have transformed the field of genetics and molecular biology. Today, DNA sequencing has become faster, more accurate, and more affordable, enabling researchers to study the genomes of other species, unravel the complexities of gene regulation, and explore the potential of gene editing technologies such as CRISPR-Cas. The completion of the Human Genome Project in 2003 was a monumental achievement that marked a major milestone in our understanding of the human genome. The legacy of the Human Genome Project continues to shape our understanding of human health and disease, and its impact will be felt for generations to come.